This presentation has been downloaded from bankofbiology.blogspot.com. In this video, I will be discussing about sex determination. In every human being or in every individual, every organism, we have two types of chromosomes. One are autosomes and another are sex chromosomes, also known as allosomes. And autosomes are the chromosomes other than sex chromosomes, and number of autosome is same in males as well as in females. Sex chromosome X and Y are the chromosome which involved in the sex determination. Henking studied spermatogenesis in some insects and discovered X chromosome. So here you see all are autosome. They are same in male as females, but these are sex chromosomes which may be different in males and females. Now we will discuss about mechanism of sex determination. And first mechanism is XX and XO mechanism. XX male heterogametic. Here XO in which gametes with X chromosomes and gametes without X chromosomes. Hetero means two types or different types of gametes are formed. So in males we have X chromosome in one gamete and no chromosome in the other gamete. And female is homogametic, means same type of gametes are formed. All gametes are with the X chromosome. So XX is the female and XO is the male. And this is exhibited by insects such as grasshopper. And next is your XX and XY mechanism. This is also male heterogametic mechanism XY, and female is homogametic, that is X only. For example, human and Drosophila. Male show X and Y chromosomes, whereas female show X and X chromosomes. In humans also we find XX and XY mechanism. Another sex determination mechanism is ZZ and ZW mechanism. Here the male is homogametic. This is very important that if the question is asked, give the examples of male heterogametic and male homogametic sex determination. So you can give the example of ZW mechanism is male homogametic, whereas XX mechanism or XXXO mechanism are male heterogametic mechanism. So male is homogametic represented by ZZ and female is heterogametic represented by Z and W. This is found in most of the birds. So in insects we are finding XX and XO mechanism, whereas in birds we find ZZ and ZW mechanism. So XXXO, XXXY mechanism show male heterogameity, whereas ZZ, ZW mechanisms shows female heterogameity. Now we see the sex determination in humans. In humans we have 22 pairs of autosomes. Total we have 46 chromosomes. Out of this 23 pairs chromosomes, 22 pairs are autosome and one pair is sex chromosome. and XX is present in female, whereas XY is present in males. So humans show male heterogamety. During spermatogenesis, we have, you see, in males 44 autosome plus XY, so it comes to be 46 chromosomes. In female we have 44 autosome plus XX, so it comes to be 46 chromosome. So in gametes, we will have 22 autosome plus X and 22 autosome plus Y. So here we find that in males, two type of gametes are formed, whereas in females, all gametes will be formed as 22 autosomes and X chromosome, and this will be giving the gametic constitution of females. If the sperm with X chromosome fuses with ovum, we get the female child or a girl child. And if the sperm with Y chromosome fuses with the ovum, we get a male child or a boy child. So, there is equal probability of fertilization of ovum with X or with Y, and sperm determines whether the offsprings will be male or female. We should always know that males are responsible for determining the sex of a child in humans. Now, a very important, sex determination in honeybees. So, in honeybees, we have two types of males. Males are drone and queens are workers. Female are queens or workers. And it is based on number of sets of chromosomes an individual receives. An offspring which is formed from union of a sperm and egg, you see this is female, this is male. So female is forming eggs and male is forming sperms. And if there is fusion of eggs and sperms, we are getting the female queen or worker. But if diploid female bee, it is producing eggs. When it is producing eggs and unfertilized eggs develop into haploid male bees, we get drone or male drone. So here we are having in honeybees the set of chromosomes are 16 and 32. Females are diploid, which are having 32 chromosomes, whereas males are haploid with 16 chromosomes. And this is called haploid diploid sex determination system. This is a very, very important question, that what is the mechanism of sex determination in honeybees? So it is haploid diploid sex determination system. Females are having 32 chromosomes and males are haploid having 16 chromosomes. In this system, males produce sperms by mitosis. This is very important because they are haploid, having 16 chromosomes. So there will be mitosis and then the gametes will be formed. And a fusion with ovum, it gives rise to female, and if it is developed from unfertilized egg from the female, it gives male with haploid constitution of genes, that is 16 chromosomes. So that was about the sex determination. Now let's see about mutation. What is mutation? Mutation is sudden and heritable change that occur in the DNA sequence. And when the DNA sequence or the gene changes, the genotype of an organism changes, as well as the phenotype. And recombination and mutation both reach or leads to variation in DNA. There are two types of mutation: frame shift mutation, which is due to the loss or deletion or gain of DNA segment. So when complete segment is lost, gained, or duplicated, we get frame shift mutation, whereas only change take place if in single base pair of DNA we have the point mutation. And the example of point mutation is sickle cell anemia. The genes are located on the chromosomes, so mutation results in abnormalities or aberrations. And chromosomes aberrations are seen in cancer cells. And the agents, this is very important. Sometimes 1 mark question, what do you mean by mutagens and what are different mutagens? Maybe 1 mark or 2 mark question. So mutagens are the agents which are inducing mutation. Mutagens are the agents inducing mutation. That may be physical mutagens such as ultraviolet radiations, alpha, beta, gamma rays, X-rays, etc. And chemical mutagens such as mustard gas, phenol, formalin, acetic acid, formic acid, ammonia, etc. So this was all about mutation.